We end on time. My name is Tom Brenna. I'm chairing this session on best practices for design, laboratory analysis, and reporting of trials involving fatty acids. And I'm going to introduce the project. We're reporting out on a project that is really took place over most of what year is this? 2018, 2017. And it resulted in a paper that is in press, and we still think will appear any day now in the American Journal of Clinical Nutrition. And so let me tell you a little bit about it before I hand it over to my co-author, Melanie Plourde. Disclosure, the project and symposium was funded in part by ILSI, North America Lipid Committee. ILSI is a public nonprofit foundation providing a forum to advance understanding of scientific issues related to nutrition and to safety and the like, and it's supported primarily from industrial membership. And Melanie has a disclosure here, which will, for her presentation, and I showed a disclosure at 6 o'clock this morning. You all saw it, right? Okay. So, so what's the issue? What prompted all this? And the question is that the problem that we hope to solve was that the heterogeneous findings in associations between blood fatty acid levels and health indicators and outcomes is confounded by differences in measurements and in reporting of blood fatty acids, but many other things even beyond that. Differences also in the way that samples are collected, in this way that samples are stored, in the way that samples are actually analyzed in the laboratory, issues surrounding design and so forth. So we're those of us who follow the fatty acid literature in any sort, really, I say. See 1 week we've got a positive finding, another time we have a neutral finding. So the question is, can this be traceable to a lack of standardization? The origins of this were and the first things that happened here was to interview experts to discuss their thoughts on this whole business. And there was real unity in the idea that setting expectations and establishing best practices would improve measurements and reporting, and with luck, improve the homogeneity of studies so that we get a little bit more clarity. A number of people were interviewed. This was before the committee was assembled, and their names are here. Other than mine, I was interviewed too. Okay, so, a core expert witness. Sorry about that. A core expert writing group was assembled. I chaired and co-chaired was Melanie Plourde. Peter Jones, Yu-Hong Lin at the NIH, Peter is at Manitoba, and Ken Stark at Waterloo. And we have presented our findings at, this would be the third conference, I think, that we're presenting findings, previous ones being the Canadian Nutrition Society and ISFAL. The process was a draft outline was made by the core writing group. Input was solicited from experts and other users. And we came up with a best practices manuscript along with supporting documents, a checklist, slides, and that would be what you're going to see today, and maybe some other things like this recording. Our outreach, the paper was accepted at AJCN on April 9th of this year. We've, as I said, presented it at a number of international conferences. The target is more or less PIs and really any stakeholder in fatty acid studies. So PIs, reviewers, journal editors. We spoke to journal editors as well. That's not on the list, but that's what we did as well. In fact, at this meeting last year a little bit. And idea was to get some idea of how we ought to proceed and the kind of information that ought to be in such a best practices document. Best practices are a much bigger thing than they were, I don't know, 50 years ago. Although there were some even historically that to some degree we've forgotten. Eventually, we would hope to have this as a tool in Equator and perhaps other places that people can access. We made a series of recommendations embedded within a narrative justification. So there's a narrative, a traditional kind of narrative review with lots of references and 22 recommendations embedded in it. We are mostly requesting is not the right word. We are mostly suggesting that rationales and reports be made, that people think about various aspects of things. Example, the blood pool, should it be red blood cells? Should it be plasma phospholipids? Should it be fasting? And we're not recommending one versus another in most cases. We're suggesting that justifications be provided in the context of the hypothesis stated for the project, so that the results can be interpreted in that light. More, we're suggesting people think about it and rather than be a loose end. We looked at fatty acid trials as fatty acids being the independent variable, so that's an intervention. When it is an intervention, study design, you'll hear about that in a moment, sample collection procedures, preparation analysis, and reporting. So that's the last one, right? Okay. So I'm going to hand it over to Melanie Plourde from the University of Sherbrooke in Quebec, who will go through some of these topics now.
So thanks, Tom. So it's my pleasure to be here to present the first recommendation. My talk is going to be more focused about sample collection, capsules, placebo, study design, and then Peter Jones will follow after that with all the recommendations on fatty acid analysis, quantification, reporting of fatty acid analysis. So the outline of my presentation will be to present the capsules, placebo recommendations, the study design, and the blood compartment. And for some of the recommendation, you will see it might be really obvious, you know, to report those things in clinical trial papers, but you would be surprised how many times these details are not reported in the paper. So that's why we want to bring a list, and then the people could just click or check the list if they have provided these details in their papers when they write their scientific papers. So before we move on, and just to give a short example, and you keep this example in your head while I walk through the recommendation. So let's start with a design, which will make it easier for having the recommendation to walk through the recommendation. So I use one of the study design I have done in my lab. And while we were doing the recommendation, I realized that some of the choices I had made for my own trial did not respond to the recommendation. So that was quite funny. And instead of using a design of other people, I decided to use my own design, so I'm not offending anybody. So, so in that trial, it was an omega-3 fatty acid and memory trial. So it was a randomized control trial. We gave a placebo or 3 grams per day of EPA plus DHA for 6 months, and we collected blood samples at baseline and 6 months later, and we performed cognitive testing before and after the supplementation. So one of the first questions usually that come out is, what type of placebo should we use? In our case, we use corn and soybean oil, 50-50 of each oil. So One thing that we made a recommendation about is the fatty acid analysis of the capsules you're using in your trial. So all the recommendations will be highlighted in yellow in my presentation. So all fatty acids in the pool or capsules relevant to the issue under investigation must be analyzed and defined. So usually when you have a clinical trial, you might provide the fatty acid profile of your omega-3 capsules. And then it's mentioned placebo is corn, soybean oil, and that's it. But for some oils, like soybean or soy oils, there's a lot of type of oils. And it doesn't give you the fatty acid profile of the placebo capsules. And this might bias your results and your design and might bias also the interpretation of your study. So we put in the paper that the independent variable must be clearly delineated. There are many different versions of plant source, so it's very important to provide a fatty acid profile of the placebo and the treatment oil. So another question we had when we were thinking about these recommendations was, what type of oil should be used in the placebo, you know? And we don't want to be really strict about the recommendation. We saw in many papers that olive oil was really popular for many years. Corn soybean oil is also quite popular. But we came to the conclusion that a blend that mimics the common fatty acid diet of the country is perhaps the best placebo, depending on the question you're asking. So it always brings back to the rationale and the question you're asking in your trial. So just think about it when you start to design your randomized control trial. What type of oil is better, is neutral for the outcome you're evaluating. So the recommendation we had with that is that control doses should be chosen to be neutral with respect to the outcome, considering the known metabolism and levels of dietary fat with respect to the hypothesis and the study population. So another part of the placebo, you know, I think that placebo is sometimes quite challenging, is that you have to provide a rationale for choosing the appropriate control oil. And whether the participants have a particular health condition, and that could bias, you know, that could be biased by the placebo. So, for example, you can have some people with cardiovascular disease, and so your placebo should be neutral with respect. It doesn't increase cardiovascular risk, and then you have good treatment because you have increased the cardiovascular risk in your placebo group. Also, the background diet of the study population should be considered, and we haven't made any good recommendation on that, but you should consider evaluating that in your trial. And in all our randomized control trial, of course, examination of the fatty acid profile in the circulation is essential for assessment of both the degree of integrity of the experimental procedure, as well as the adherence level of individual participants. And for that, we come to the recommendation that the rationale of the choice of the blood pool should be stated and justified with respect to the biological research question, taking into account logistical issues with respect to study design. So that come to the question, what blood pool should I look at, my fatty acid compliance? And Ken Stark published the global map study last year, or 2 years ago, I don't remember. They reported that there are 4 main blood pools that are reported in studies. So there's plasma total lipids, plasma phospholipids, erythrocytes, and whole blood. So this doesn't mean that these are the only blood pool, but these are the most popular ones that are reported in the different trials. So let's look a little bit closer to some of the differences. So this is a study where they provided EPA plus DHA for 4 weeks, and it shows a different pool. So in yellow you have plasma, in blue you have whole blood, and in red you have the red blood cells. So on the left you have the EPA levels increase and decrease, and on the right you have the DHA levels. So if I use the arrow, yeah, that's working. So they provided the EPA plus DHA supplementation for 4 weeks, and then they stopped the supplementation and they look at the washout of the omega-3 in the different blood pools. So you see that within 4 weeks, EPA levels increase very much in plasma, whole blood, and red blood cells. And then when you stop the supplementation, it goes back to quite a baseline 4 weeks later. But look at the DHA levels. So the DHA levels increase in plasma and whole blood 4 weeks after the supplementation, but not in red blood cells. So if you have a trial of 4 weeks and you want to measure compliance by evaluating DHA in RBC, this might not be the right marker, for evaluating compliance. So you have to take in consideration when you choose a blood pool, what is the duration, what time, how long do you have to supplement to expect an increase, what type of increase you should expect, and compare your results to previous study. If you look at finger pricks, this is a study that was published in 2009. You see that the EPA and DHA levels increase a lot within 4 weeks, and then when you stop the supplementation, there's a clear drop of the EPA plus DHA level in the finger pricks. But it's mostly driven by the EPA level, which increase a lot within the first week of supplementation, whereas DHA tend to increase smoothly. So really, when you have to pick up a blood pool, you have to consider the intervention length that should be rationalized based on the hypothesis, and you have to look at the fatty acid pool that is required to evaluate your fatty acid increase, because there are various time periods for recalibration depending on the pool you're choosing. And studies of short duration will fail to test a hypothesis requiring long-term equilibration times in the plasma or blood. So you might consider that in your trials. Another recommendation we have is that the levels of particular or some fatty acids, such as omega-3 fatty acids, are modified in specific population and our blood pools. And these changes should be taken into account when interpreting the results. So let's, maybe some of you are familiar with the global map paper. So what this group performed, they looked at 298 studies that were included in that paper. And they reported, they had the fatty acid profile in different countries. And so they kind of do a map of the fatty acid profile in the red blood cells, the EPA plus DHA in the red blood cells across the world. And so this is for the red blood cells here. So in red, you have EPA plus DHA total fatty acids in erythrocytes equivalent less than 4%. And orange, you have, it's between 4 and 6%. In yellow, it's between 6 and 8%. And in green, it's higher than 8%. So you can see that around the world, depending on dietary and lifestyles and everything, and dietary intakes of omega-3, you have different levels of fatty acids. And it varies a lot between the different countries. And if you look at the different blood pools, so in plasma total lipid, you have the same map, but with different ranges. So for example, plasma total lipid, it ranges from less than 2.9 to higher than 5.2. Plasma phospholipids, less than 3.8 to more than 7.6, and so on. So really, depending on the blood pool you're choosing, you have some ranges where in your country you should expect the fatty acid profile to be within. So just keep that in mind, and if you want these ranges, you can look at the paper of Ken Stark that was published in 2016. So another recommendation is that dietary fatty acid exposure and implications for well-established principles of fatty acid metabolism must be taken into account for interpreting the diet-blood relationship. So if you eat a lot of, for example, saturated fat, maybe you won't increase that much your saturated fat in your plasma. So the dose response of EPA plus DHA is much greater than the saturated fat response. Therefore, sensitivity is greater for EPA and DHA as a rough rule. So some pools and fatty acid do not respond to treatment, and it's quite challenging sometimes to monitor compliance of the placebo for that reason, because the composition of the placebo does not translate into increases in some fatty acids in the blood pool. So it's quite challenging at that point, and we don't have any good good recommendation for that, but you just have to consider that when you interpret your data. So for example, for the expected range of increase, so these are, this is a paper by Pedersen showing the relationship between EPA plus DHA in total in erythrocytes, venous whole blood here, and plasma phospholipids. So you have, oops, sorry. So you have the level here on the y-axis, and here you have the EPA plus DHA intake. And then you can see that it's quite linear under 1 gram per day of EPA plus DHA for the different pools, so erythrocytes, venous whole blood, and plasma phospholipid. So from this paper, they mentioned that the EPA plus DHA increased from 85 to 95% for every gram of EPA plus DHA consumed over a negligible intake baseline. So this is a rough rule, but it's just to give you an idea of what you should expect. So all fatty acid, another recommendation we had is that all fatty acid intervention trials should report at least one measure of fatty acid concentration, not just relative percentage, as within a specific biological compartment, so as to enable analysis of both an intention to treat and a per protocol analysis. So if we come back to the research design I presented you at the beginning of this talk. So just to, maybe some of you are not too familiar about the difference between intention to treat and the per protocol analysis. So we just want to give you a couple of examples about that. So if you say in our trial that you want to increase DHA status by giving a supplementation of omega-3, and because the increase in DHA status in the blood will improve cognition. So this is a per protocol analysis, because to evaluate that, it will require that you do the fatty acid analysis in the plasma, okay? And then you can measure actually the increase in omega-3 in the blood and then connect that with the cognition test. Whereas the intention to treat is, I would say to my participants, I'm prescribing you to take omega-3 fatty acids, and I think it will improve your cognition. But then it's just a prescription. I'm not checking if you're taking the pills. And then you have the scenario C here, where, for example, those that were only compliant to the prescription, they got the better cognition. And then for evaluating C, you need to do the fatty acid profile. So if you do the intention to treat analysis, and you want to evaluate your research question as a per protocol, then you just do the fatty acid profile in that specific case. You do the fatty acid profile in the plasma, and then you can convert your analysis. I know it's a simplistic way of presenting it, but it's just to give you an idea of how you can present both data in the same paper. So this will bring us to another recommendation that we made, is that intervention-based changes in circulating fatty acids should be compared with previous literature reports to establish that they fall into expected range. So omega-3 fatty acid levels in the circulation, in the circulating compartment, are an indicator of adherence, so you can use that as an indicator. And of course, there's genetic risk that can modify the response to the supplementation, and you can discuss that in your paper. But as a rough rule, if you increase omega-3, you increase your omega-3 in the plasma when you take the omega-3, and then if you got non-responders, you can mention that in the paper, but at least you consider those points. And in all randomized controlled trial, adherence, it should be indicated how you measure adherence, compliance to your treatment. So is it just on pills, or did you do some fancy stuff to evaluate compliance? You just have to report that. So what is an expected range of increased EPA plus DHA following a supplementation? So I'm going to present you these data here. This is a paper that was published by the group of Peter Jones, where they control-fed, it was a control-fed feeding trial where they provided different type of oils, and they provided canola DHA oil. It was providing roughly 4 grams per day of DHA in this oil. So we know that compliance was there because it's control feeding. So they come to the research center and they were fed, and then we know that they took the oil. So for that trial, 4 grams per day for 4 weeks translated to roughly plasma DHA in total lipids increased by 2-fold. So as a rough rule, that's the type of increase you should expect if you give 4 grams per day. Of course, there's difference in the trial that can bias a little bit this response, but it's just to give you an idea of the type of response you should expect. So also reports should outline medical, physiological, and behavioral conditions that may influence fatty acid target outcomes, and discuss steps taken to minimize their effect. So if you're studying pregnancy, you should expect that your results will be different from women that are not non-pregnant in your study. So just to report these things. And I want to give an example, because I'm studying a lot the biology of aging and fatty acid metabolism. So one thing that we've seen, these are unpublished data from my lab, is that during aging, there's an increased EPA level in the plasma of older adults compared to young adults. And there's kind of a U shape for DHA and a higher level of arachidonic acid. And this is without supplementation, and this is not because older adults consume higher fish in this study. So it's just like there's a lot of paper showing that during aging, there's an increased level of EPA in the plasma. It's almost twice as much as the one in young participants. We don't really know why, but you know, this is what happened. This is the situation. So if you use a wide range of participants with a right range of age in your trial, then you should consider that maybe the baseline data of your participants would be different according to age. So with that, I'll leave the floor to my colleague, Peter Jones, and then we'll take questions after both presentations, because since you may have some questions about analysis and everything that maybe Tom will answer, or at least will provide the recommendation. But after, we'll do a panel of discussion, and you could ask your question on my or Peter's presentation. Thank you.